All right, so today's topic, we go into cells. Now, when we talk about cells, there are a couple of different things that, not just a couple, but quite a few different things that we want to keep in mind. Now, if I took any one of you and I cut you in half, and I take one of those halves and I cut it in half, and I take one of those halves and I cut it in half, and I continue and continue and continue, I'm going to end up with the smallest particle possible where I still have life. And that is going to be the cell. Now, can I get smaller than the cell? Yes, because you can get down to the level of the organelles. And can I take these organelles and cut them in half? Yes, I would end up with atoms. And again, if we take an atom and we start dividing it, we get down to the protons, neutrons, and electrons, our subatomic particles. But once I go any further down than the cell, I no longer have life, and that's the difference. And so therefore, the smallest possible thing that exists that still has life is what we refer to as the cell. Now that, in essence, gives, what we would say, gives creation to cell theory. Now, cell theory is basically made up of three statements. Typically, when we say, oh, what is this particular theory, we talk about a belief in terms of if this happens, then that will occur. Well, cell theory is not exactly the same. Cell theory is just basically a theory that states three facts, if you will, that we have regarding cells. Now, the first one is that all living things are made up of cells. The cell is the basic unit of life, and finally that cells come from pre-existing cells. Now, based on what I was just speaking to you about just a second ago, all living things are made up of cells. When we discovered that the cell is the basic unit of life, that gave us that very first part to cell theory. And from there, we later discovered that all plants and then all animals are made up of cells. And that gave us the second. And then after that, we came up with the idea that the cell basically comes from a pre-existing cell. All three components together is what we refer to as cell theory. In the mid-1600s, if you're going to associate a person with cells, it's going to be that name right there, Robert Hooke. Robert Hooke in 1665 did his earliest work with a slice of cork. He took a slice of cork, which is a material from a plant, and he cut the most thinnest slice possible. And he observed that under the compound light microscope. And he discovered what he named cells. He named them cells because he spent some time in a monastery, and they reminded him of his cell, his living quarters, which were called cells. So he called them cells. And he thought they were basically empty little spaces inside of that slice of cork. Later, we realized that they're not empty, that they're actually organelles, functioning parts inside of these cells. But if you're going to make an association with cells, make sure that it is with the name Robert Hooke. He is also very highly associated with microscopes. Okay? He did some of his best and biggest work with the microscope, and also really brought the use of microscopes to the forefront of science where it is today. So he basically comes up with the idea that these cells are the major part to the three ideas for cell theory. Now, Anton von Leeuwenhoek later takes a sample of pond water, and based on that, he sees small living things lurking around inside the water as well, again, under the microscope. And he basically makes the discovery of what today we call bacteria. He knew that in pond water there would be bacteria, and again, back in these times, it was very common to drink that kind of water. So he took a sample from the oral cavity of people and saw other types of bacteria in that as well. And he drew them, and this is what they basically look like. So he saw these living organisms that were fully functioning, independent living organisms. And again, that hence goes even further to solidify the component of cell theory that says that the cell is the basic unit of life. Years later, after von Leeuwenhoek makes that discovery of the cell being the basic unit of life, Matthias Schleiden concludes that plants are made of... All right, so as I had mentioned earlier, Schleiden was the one that discovered that all plants are made of cells. And then a year later, Schwann discovers the same thing in animals. So basically, if Leeuwenhoek realizes that bacteria is living and made of cells, and Schleiden discovers the same thing about plants, that plants are made of cells, and that it has been determined that the cell is a living organism, and then Schwann discovers the same thing about animals, then at that point we've basically covered all living things, plants, animals, and anything that falls into that scope of bacteria is considered to be living and completely comprised of cells. So that gives us our biggest second part of cell theory, that all living things are made of cells. And then finally, about a decade or 15 years later, Rudolf Virchow concludes that cells can only be produced by taking a pre-existing cell and dividing it. Now that idea had already originated 50 years prior, but as many scientists do their work throughout their whole life, and sometimes even die of old age never seeking that answer that they wish to find, someone else somewhere out there is going to pick up on their work. And that's basically what he did. And so 50 years after Lawrence Oken comes up with the idea that cells divide to make a new cell, Virchow actually gets the evidence that he needs and makes the third part to cell theory. So between all of them, and it takes over 200 years to basically come up with cell theory, we have our three major parts of cell theory. The cell is the basic unit of life. All living things are made of cells, and cells come from pre-existing cells. Now all this work with cells has to give us an indication toward what we use to learn about them, which is the microscope. Now here in this class, we basically use the compound light microscope. But you do need to have a little bit of an understanding about what is an electron microscope when we go to look at things that the compound light microscope doesn't quite give us. The compound light microscope basically works with lenses in two different locations. Light, basically a source of light shining from underneath, which basically goes right through your sample. Now that basically means that you're going to be able to see the sample somewhat see-through, so it gives a kind of a hard time trying to see what's inside of something like a cell. The compound light microscope is a great instrument to use, but obviously, like anything that's simple, it has its limitations. It has its pros and its cons. The first set of lenses, going back to the previous slide, we call the objectives. And when we go to work with a microscope here for the first time, you'll be able to change from one objective to another. They're very delicate, very expensive to use, and they work rather well. And they allow you to see the sample here on the slide. And then you have a second lens, which is going to be your eyepiece. And compound light microscopes use the light source reflecting off of mirrors. And they basically reflect light off of these mirrors so that you're able to see from all sides. They diffract the light, if you will. An electron microscope does not. There is no diffraction there. It's basically shooting electrons either through or at the image. And I'll get more into that in just a minute. So what are some of the problems? Now again, I told you that because of the fact that the compound light microscope uses diffracted light, it can only magnify something about 1,000 times its size. Now that may sound like a lot, but in reality, when we're looking at something really small, like dealing with cells, 1,000 times isn't really all that much to see certain things. One of the other problems with it is that the light basically makes whatever you're seeing on that slide transparent. So you're going to be able to see right through it. So you're going to lose detail when you use a compound light microscope to be able to see things. And the fact that the cell itself becomes kind of transparent, that means that smaller objects inside of the cell you won't be able to see. So we have to go through a technique called staining. And that's where we apply a particular dye made out of certain elements that are basically going to attract to certain things. I'll talk to you about fluorescent dyes in a second, but right now just typical blue stains and dyes, which is what we're going to use here in class, will basically attach themselves to particular molecules that are located in the cell. And then through this is how you're able to see the parts inside. Otherwise, if that wouldn't occur, you wouldn't be able to see inside. Now, going back to what I had mentioned a second ago, the fluorescent dyes, there are certain fluorescent dyes that attach themselves to specific molecules, and that's a particular type of microscope. It's called the fluorescence microscope. Now with that, you're able to see, even in a cell, you can actually see things moving around inside of that. So whenever you see that in the movies and so forth, they see one cell maybe come into contact with another cell and so forth, that's through a fluorescent microscope. A compound light microscope can never give you that kind of imagery. Again, this is also very expensive, and only certain institutions would even have access to these kinds of instruments. Now, both of those are very limited when compared to an electron microscope. There are two types of electron microscope, TES and SES. The TES is what we call a transmission electron microscope, and the SES is a scanning electron microscope. SEM, I'm sorry. Both of them basically disperse electrons to the slide of whatever it is that you're looking at. But since light refracts in all different directions, you can't have electrons do the same thing. So they work through what we call a vacuum. If you see a picture of an electron microscope, whether it's a transmission or scanning, you'll see a rather large vacuum where the electrons are basically shot at from the top down to the slide. Now the major difference is a transmission electron microscope. The electrons go through your sample. So you basically, in order for that to work, you have to prepare the slide in a particular way. Now, one of the ways to be able to do that is it's going to give you, for one, a much higher resolution. But in order for you to get that resolution, you have to remove all the water from the sample. And then what you're end up getting at that point is a flat two-dimensional image, right? That's a transmission electron microscope. The electrons have to be able to pass all the way through the cell and the tissue, and in order for that to happen, that tissue needs to be cut really thin. Hence, you have to remove the water out of that. So in an electron microscope, you can never see what? A living sample. You're never going to be able to see a living sample. Because of the preparation involved in it, you're only going to be looking at a flat two-dimensional image of something that was once living. But you're able to see cell structures, you're able to see protein molecules after they've formed. And that's something that takes a high amount of magnification compared to, for example, a compound light microscope. Now, a scanning electron microscope, the electrons actually bounce off the surface of the object. And just like radar, for example, what do you end up getting? You end up getting a more of a 3D type image. You understand how radar works on the bottom of a ship? When it wants to get an image or a layout of the topography of the ocean floor, you're getting that sound wave that basically, will show you the ground is higher here, lower here, and so forth. And some of these can actually be so sophisticated where they'll actually show you details of small crevices and rocks, something that might be moving across the ocean floor and so forth, and you're basically just getting a 3D image of that. This scanning electron microscope works basically in the same way. So the electrons bounce off the surface of the object, and it gives you that 3D image of whatever it is that you're looking at. There's a very popular image in Google about bed bites, and it's green because it was an electron microscope was used to get that image. And bed bites, or bed bugs, sorry, are really tiny, microscopic. If you look at them, it almost looks like nothing. It looks like just a tiny speck of dust. But if you ever get a chance, take a look at that image through Google, and you'll actually see eyes, a jawline, teeth, claws, and so forth. It almost looks like a monster, if you will. And these things are, when you look at it on the tip of your finger, you're seeing a speck of dust. And you would feel nothing, but lie down in a bed that is full of bed bugs, and you're going to wake up with those red sores all over your body. So actually, they're very small, but they are large enough where we can see these images. And one day, whenever you get a chance, take a look at that and you'll see what I'm talking about. Now, electrons can easily scatter. So like I was mentioning earlier, electron microscopes use a vacuum in order to get a good image of what you're looking at. Like I said, the water needs to be taken out before placing it in the microscope, and so therefore you would only be able to examine non-living cells and tissue. Now let's talk a little bit about prokaryotes and eukaryotes, some of the similarities and differences there. If I asked you what is the major difference between a prokaryotic cell and a eukaryotic cell, the first thing that should pop into your mind is this structure right here, which is the nucleus. Now, prokaryotes do not have a nucleus, or rather their genetic material is not in an organ-bound nucleus located in the center of the cell. Eukaryotes do have a nucleus. By far, that should be the very first thing that pops into your mind when asked what the major differences are between a prokaryote and a eukaryote. One of the easy way to remember that is the prefix. You know how words have more than one meaning? Well, prefixes are applied to the same standard. a prefix can have more than one meaning. The prefix "eu" means true, and karyote means nucleus. So a eukaryote is a cell which has a true, which has a true nucleus. The prefix "pro" means before. Well, before what? Before having a true nucleus. So a prokaryote means before having the nucleus. This also going a little further back to when we were covering evolution, again is one of the major factors that gave us the idea that the prokaryotic cell came first before the eukaryotic cell. Prokaryotic cells are much smaller than eukaryotic cells. That should be, one of the things that should kind of indicate to you that the prokaryote doesn't have a nucleus. But it does have genetic material, obviously. So if it doesn't have a nucleus, where would the genetic material be found? Well, it's basically found, sometimes it's found in what we call this nucleoid region, and it can also be found throughout the cell wall, and it can also be found in the cytoplasm, floating around in the cytoplasm as well. So the nucleus, for the most part in a prokaryotic cell, is everywhere, where in a eukaryote, our genetic material is primarily located here in the nucleus. So again, prokaryotes, no nucleus. Eukaryotes has a nucleus. Prokaryotic cells are much more simple than a eukaryotic cell. Again, this is not true to size, but they are much smaller. There is a motility factor that is a big difference in them. But some of the similarities is that they do have their genetic material. Both have ribosomes, and when we get into DNA, I'll talk to you about two terms, transcription and translation. And the ribosome is very important for that, because without that, you can't duplicate that genetic material. And both have that cytoplasm, which is that substance, that gel-like substance on the inside that basically keeps all these organelles from moving around all over the place. Their difference in size. The prokaryotic cell much smaller than the eukaryotic cell. Again, I can't stress enough the major difference, though, is being that nucleus. I want you to always remember that the prokaryotic cell, no nucleus. The eukaryotic cell has a nucleus. I talked to you about motility a few minutes ago. Prokaryotic cells move, the ones that do, move in one of three ways. First, they could have what we call cilia. You can barely see it here in the image, but it's cilia is basically these microscopic fiber-like hair structures. And just like a centipede uses them to move across, the cilia form that peristalsis type movement underneath and allow the cell to move. And the other way that they could move around is through this tail. We don't call it a tail. Anyone know what it's called? It's called a flagella. And this flagella, which is kind of like that whip-like tail, allow them to move. And finally, very few do this, but they also can move through what we call contraction, like the earthworm, where it contracts and releases, contracts and releases as a whole, and that allows it to move across. Again, remember, we're primarily talking about these things being in a liquid type environment. So movement is not anything that's really difficult for prokaryotic cells if they've got the ability to do so. Now, eukaryotic cells are, again, much larger. When we think of eukaryotic cells, think of plants, animals, fungi, protists, things like that. Whereas to the prokaryotic cell is primarily, excuse me, bacteria. They're more complex. Since they're larger in size, part of the reason for that is they've got twice as many organelles inside of them. If you see the structure of the prokaryotic cell, there's barely any organelles in there. Whereas to the eukaryotic cell is full of a bunch of different types of organelles that perform all these different functions. So they're much more specialized than the prokaryotic cell. Now let's talk about these organelles for a bit. I want you to understand first which organelles can be found in either the prokaryotic cell, the eukaryotic cell, or both. And if they are found in the eukaryotic cell, I want you to be able to identify whether they are found in plant or animal or in both. And then this column here is going to tell you basically what they do, what their function is. So first we start out with the most important one, the nucleus. Imagine if you go to the store and you buy a piece of furniture. Now you're not there yet, but as an adult, you someday are going to buy a piece of furniture like a wall unit or a table or a chair or something, and it's going to come unassembled. And you're going to have to assemble that. Well, when you open that box and you want to start assembling that piece of furniture, what is the one thing that you are going to find most useful? The set of instructions, that manual. And without it, maybe you would be able to put it together, but chances are that you probably what? Would not. You would, and at the very least, maybe you would be able to, but you would find it very, very difficult. Well, the nucleus holds that set of instructions for the cell, your genetic material. It tells the cell when it can divide, how it can divide, when it does divide, what are the reasons it does, and when it does divide, how the new one is supposed to be, how the new one is supposed to look, how the new one is supposed to divide when the day comes that, or the time comes that it is supposed to divide. So basically everything is there. Without those set of instructions, it basically renders the cell useless. So the nucleus is very important. It holds that genetic material for the cell. The nucleus also allows the RNA to be transported in and out. And again, I'll mention, we'll talk more about that when we do DNA. But I want you to understand for right now one thing. That genetic material, that DNA, that cannot leave the nucleus. Okay? It always stays inside the nucleus. Now the nucleus is found in eukaryotic cells, not prokaryotic cells, and in both plant and animal. Both plant cells and animal cells obviously have a nucleus. Then let's talk about the ribosome. The ribosome is also found in both the prokaryotes and the eukaryotes. And when it's found in the eukaryotes, it's also found in both plant and animal. And this is where proteins are made. Remember when we talked about the 4 levels of organization? Primary, secondary, tertiary, quaternary. Those levels of organization talked about what? How a protein is made. The 4 things that need to occur when a protein is made. Those codons of amino acids that get together to make these chains, that chain is going to continue to fold and so forth, and eventually this chain and that chain all combine to make that 3D quaternary structure known as that protein. That happens here in the ribosome. Okay? Proteins are made in the ribosome. The mitochondria is eukaryotic. Okay? Prokaryotes do not have a mitochondria. In the eukaryotes, it is found in both plant and animal. Now, what's the mitochondria primarily responsible for? When I say the word mitochondria, there is one word, starts with the letter E, that should pop into your head. And that word is energy. The mitochondria is where cellular respiration occurs. And cellular respiration is basically what produces ATP. It's known as adenosine triphosphate. It is the energy for the cell. Chloroplast. Now chloroplast has that prefix chloro. Okay, and that right there should basically tell you that that is a plant organelle, not animal. But it is eukaryotic. Now there are some bacterial cells that go through photosynthesis, but primarily those are eukaryotes, not prokaryotes. So those bacterial cells that do go through photosynthesis are eukaryotic in nature. The chloroplast is where photosynthesis occurs. When we talk about excuse me, when we talk about plants, we're going to talk about how the leaf have these small little stacks, okay? And in these little stacks, or these round little objects called thylakoids, that's where basically photosynthesis takes place, the light-dependent structures inside of the leaf. And when photosynthesis occurs there inside of that plant cell, it occurs specifically in the chloroplast. Okay? It's the one structure that gives the leaves that green type color. Then we have the ER. We have two ERs, the endoplasmic reticulum. We have a smooth ER and a rough ER. The difference between the two visually is very simple. Smooth means you're not going to see any small little bumps on it. And the rough ER, you will see these small little bumps on it. Those little bumps are ribosomes. Okay? So we find ribosomes in the rough ER, not in the smooth ER. Now I mentioned to you about how ribosomes are what make proteins, necessary to make proteins. Well, where do they do it? They do it in the endoplasmic reticulum. It's the highway of the cell, the site of the protein and lipid synthesis. So these ribosomes basically at the endoplasmic reticulum, at the rough ER, are going to fold to increase the surface area, and that allows them to make more proteins and lipids. Okay? They are eukaryotic, not prokaryotes, and they are both found in both animal and plant cells. The Golgi body, also known as the Golgi apparatus, is eukaryotic and found also in plant and animal. And that's basically the one that sorts and packages the products inside of the cell. In an upcoming slide, I'm going to show you the one called Cell City, where all the different parts inside of a city do different things. Well, think of the Golgi apparatus as like the post office of the cell. It's the one that's responsible for packaging and transmitting or moving around certain things within the cell, sending them to where they need to go. Lysosomes are eukaryotic and also found in both plant and animal cells, and they're the ones that basically break down the waste products inside of the cell. I tried to think of anything in terms of like, you know, an association. Remember, I learned through association. If you can associate anything with lysosomes, there's a cleaning product. The name escapes me now. Lysol, I think it is. And that does what? It breaks down bacterial agents on a surface that you clean. So if you can kind of associate that with the lysosome as being the thing that breaks down waste products inside of the cell, that might help you somewhat. The cell wall, obviously not found in animal cells, only found in plants, but it is also prokaryotic and eukaryotic. So prokaryotes do have a cell wall, and eukaryotes also have a cell wall, plant cells, in other words. Remember, where we have mobility and our eukaryotic cells in animals have that mobility, we don't need the cell wall as that added protection. But plants who do not have that mobility that we do, they do need that added sense of protection. So they have that cell wall, that hardening structure that creates that outer surface of the plant cell. It protects the plants, and some prokaryotic cell, it gives them support. We find cellulose inside of the cell wall, which is that what? That sugar that they use for energy and for protection around the cell. It gives it its strength as well. And then we have the vacuole. I mentioned here briefly about how the lysosome breaks down waste product. Well, the vacuole is kind of like the garbage can, if you will, of the cell. Now, in a garbage can, we store our waste product, but it also stores water in there as well. It is eukaryotic, and plant cells have a vacuole, not animal cells. And they're going to have one central vacuole, one large vacuole, and actually that takes up the majority of the plant cell. That's where the majority of the plant's water is stored. And it's also going to have some smaller vacuoles as well that are going to store water and waste in there as well. The vacuole supports the plant cell because it occupies the majority of the inside of the cell. That basically exerts a pressure on that cell wall. And that pressure is what basically maintains that cell wall, or rather that cell, to keep its shape. Anytime you see a cell of a plant, you always see it as like straight lines, if you will, where an animal cell can be more wavy in nature. But the reason for that is the pressure being exerted on that cell wall. The cytoplasm is that gel-like structure that is inside of both prokaryotes and eukaryotes, and in terms of eukaryotes, both plant and animal. Think of it as like that jelly substance on the inside of the cell that doesn't allow the organelles to bounce around all over the place. The cell membrane, we don't have a cell wall, but we do have a cell membrane. And even plant cells who have a cell wall also have a cell membrane. The membrane is important because that's what basically determines what goes in and out of the cell. When water passes in and out through a cell, water can carry certain proteins and nutrients and so forth in it. And that cell might want them. So the membrane has to be the structure that opens up, allows them to go through and stay inside, or continue and pass through the other side. When we go through our next topic, osmosis and diffusion, we'll talk to you, or I'll talk to you about what we call hypertonic, hypotonic, and isotonic. And the cell membrane plays a large part in establishing osmosis and diffusion. Now, because I told you that water goes through it, that's what we refer to as being semi-permeable. Okay? And your cell membrane is also known as what we call a lipid bilayer. Lipids are what? Fats and oils. That means that if your cell membrane is made, or is also called a lipid bilayer, that means that your cell membrane is made of fats and oils. Many times when you go to the doctor and the doctor tells you, yes, you have to reduce the amount of fat and oil out of your diet, but they'll never tell you to completely, entirely take it out. Because we need small amounts of fats and oils in our diet. Why? Because our body uses it to what? To make that cell membrane. I believe in an upcoming slide I'm going to talk to you about how those lipids line up to make that membrane. But I basically already explained it back when we did evolution, when I talked to you about the protenoid microsphere. Remember the protenoid microsphere? How it basically lined up all the heads of the lipids facing, some facing the outside of the cell, some are facing inside the cell, and the heads had a what? A hydrophobic tail which faced the inside. And so the head faced the outside, which is the part that, touches water, and the tail faced in. And then on the inside layer, because it's a bilayer, and bi means two, so the inside layer, the head faced the far inside of the cell, pointing the tail to the inside as well. And so you get that. I got a picture coming up now in a few slides that shows that lipid bilayer. But it is selectively permeable, means that it basically can determine through these certain pumps that it has, it can determine what goes in and out of the cell. The cell membrane is the one that determines what goes in and out of that cell. And I'll talk to you a little bit about what's called passive transport and active transport, but it's very self-explanatory. Passive transport means that what? No energy is required. So it's passive. So whenever things are going to pass through that cell membrane, through what we call passive transport, there's no energy required. You basically want to move particles from an area of high concentration to an area of low concentration until an equilibrium is established. Once an equilibrium is established, we say that the cell has become isotonic. Okay? And when that happens, then the cell membrane basically stops. And so if one goes in, then what? One has to come out. And if one goes out, then one has to go in, and so forth. So it tries to keep that balance once it has established being isotonic. Hypertonic cells can either be hypertonic versus their environment, which is hypotonic. But again, when we get to osmosis and diffusion, we'll get more into that. Right now, just remember that the cell membrane is what determines what goes in and out of the cell. And then we have cytoplasm. Is cytoplasm a duplicate? Yeah, it is. So I already talked about that. This is that picture that I talked to you about the cell city. It isn't interactive. It was where I got it, but I just basically wanted the picture to basically show you all the different things where cell city is the entire cell. So you have storage tanks of food and water. What are those? The vacuoles. The vacuoles. Now remember when I told you mitochondria, we think that one word that starts with E, which is our energy. This would be our mitochondria. We have our construction site where things are made. So something is going to be made here. What are we making here? Typically proteins, right? And then those proteins need to be, they need a source of transportation, if you will. And then they're going to be transported. Now see these highways. Well, we're going to get there now, but I'm just basically giving you the idea where something is going to be transported through these highways to either a storage facility, to be processed, so that after processing it can go to be used as energy. Maybe it goes down here for regulatory purposes, to be broken down, and waste can be discarded. Obviously here the walls of what goes in and out, you know what that is going to be. So look at these numbers and we'll revert to this back and forth. Let's go to number 1 first. Number 1 is our construction site. What is our construction site? the ribosome. Okay, what does the ribosome make? The production of protein, right? So it builds these new structures. Now after, it needs that transport company, what we call the endoplasmic reticulum. Remember that we have two. We have the rough endoplasmic reticulum and the smooth endoplasmic reticulum. So the transportation company is the ER, which transports the proteins made by the ribosomes. And then we have our power plant, which is the mitochondria. After these proteins are going to be sent to the chloroplast, which is our processing area, they can then go from here off to be utilized as energy in the power plant. Whatever doesn't go to the mitochondria from the chloroplast can also go to other parts, any other part of the cell. The waste that needs to be disposed of, remember, if you're like me and you learn through association, that lysol will give you that first part of that word, lysosome, to break down the waste product. And that would be our number 5 right here, where the waste disposal plant basically breaks it down, anything that's not needed, to then either be sent off to storage, and eventually that vacuole is going to release it out to excrete out of the organism. Number 6 is that city hall. And city hall is what we refer to as what? The nucleus, where basically everything is determined. I gave you that word of regulation. Well, the nucleus is what basically regulates all those other jobs in that cell. And when I say regulates, I mean determines. Okay? The vacuole was the storage tanks where food and water are going to be stored, and that cell membrane or cell wall is going to be that gateway of what goes in and out of the cell. Now these are all the major organelles that we need to know in a cell and what they basically do. Now, let's talk a little bit about that phospholipid bilayer that I mentioned earlier. Remember I told you about how the lipid has a head and a tail. The head is okay with water. So there's a term for that. We call that hydrophilic. Hydrophilic means you're okay with water. The tail is the complete opposite. The tail is not okay with water. So that means it fears water. So it is hydrophobic. Okay, the hydrophobic tail always faces the inside, as it does right here, and the hydrophilic heads face the parts that do touch water. These are lipids that are going to form in a large chain. We call it a fatty acid chain. And they need a glycerol, okay, and they need a phosphate group. And when they get this, they form that phospholipid. That's basically the structure of the cell membrane. The heads contain phosphorus and nitrogen. Now, why is that important? Because that gives them polarity. And what did we say about the polarity of water? Now let's do a little review of properties of water. What did we say about the polarity of water? Remember that a water molecule has the oxygen and the 2 hydrogen. And basically remember that it's polar, which means what? That those electrons are what? Not going to be shared equally. Remember my example of you play with it for 5 minutes, but then before the 5 minutes, oh no, can I give it back to me? And I play with it for 5 minutes, and then you give it to the other one, like that. Well, those electrons spend a majority of time going around oxygen more time than they do going around hydrogen. So because of that, they are what we refer to as being polar. That gives, at the same time, a neutrality to water, but it also gives it a slight charge. You take that water molecule and it will be negative on one side and positive on the other. Now because it is negative on one side and positive on the other, there is your neutral. So yeah, water is basically neutral, but when you break it down to that level, the level of the molecule, it's neutral because it is positive on one side and negative on the other. That gives it its polarity. So water molecules are able to what? To each other. They're all able to basically stick to each other. Now because of that, that means that the head of the lipid is going to contain phosphorus and nitrogen, which makes it polar. Therefore, since water is polar, then the hydrophilic head is going to be attracted to water. So it's okay that it comes into contact with water. Now the tail, on the other hand, does not. The tail is basically just part of the major part of the structure part of that fatty acid. So there is no polarity there. And if it came into contact with water, it would basically, since water is going to have a stronger polarity than the lipid, it's basically going to separate it. Which is the reason why, one of the reasons why, when you pour lipids like oils into water, they don't what? They don't mix. They don't mix. Okay? They lack that polarity between the two. So again, hydrophilic head, hydrophobic tail, tails face to the inside of that lipid bilayer so that they don't come into contact with water. But water, we said that the cell membrane is semi-permeable. So if it's semi-permeable, that means that what? Water has to pass through it. So the tails do come into contact with water, but then these tails, see their shape? It allows them, with one small little movement, it will allow them to what? To basically push the water right through. So it's kind of like that icky factor, if you will, that once water goes into the membrane, it just wants water to just, get out and continue to pass all the way through. So I'm giving you this picture. This is the lipid bilayer. Your head's facing, let's say this is the outside of the cell, this is the inside of the cell. So here's your lipid bilayer. Your hydrophilic heads on this side come into contact with water. The hydrophobic tails on the inside doesn't come into contact with water. And the same thing on this side. Your tail faces in, your hydrophilic head faces the inside of the cell, which also has water. Now, when certain particles are going to pass through this membrane, there are these things called protein channels or protein pumps that allow proteins and other particles to go through the membrane. And they are, remember that word, selective. You see this protein channel here? He basically accepts that particle and basically reads it as, yes, I want this to go into the cell, or I want this to come out of the cell. And so then it will take it, move it across the lipid bilayer, and basically pump it out. Or here, when the cell is trying to establish becoming isotonic. Let's say, for example, there are more particles on the outside of the cell, or rather I should say, there is a higher concentration of particles on the outside of the cell than there are on the inside of the cell. We would say that the cell itself, what did I say, that there's less inside, right? We would say that the inside of the cell itself is hypotonic. So there's a smaller concentration of particles inside than outside. So it is hypo. Hypo means below. The outside of the cell, in turn, would be hypertonic. The prefix hyper means above. So what is the cell going to do to try to establish becoming isotonic? If there are more particles outside than there are inside, you know what it's going to do? It's basically going to allow particles in and closing right away so that nothing gets out. But I'm showing you both in this instant because it could be hypertonic on the inside of the cell versus being in a hypotonic environment. So it works both ways. But this protein pump basically is going to move things selectively, but it's going to move things in or out of that cell until it reaches that balance. Or maybe the cell, you just ate something and the cell's looking for particular proteins, nutrients, particles that it needs, that it lacks. Maybe even just, some of the things in the water itself or water itself. It will allow the membrane to let things to pass through and then keep them trapped inside. There's a really good video when you go to do the gizmo on this that you're going to be able to see osmosis and diffusion where it basically particles pass through and it talks about active and passive transport, where through passive transport the particles just basically pass through the membrane and they can continue passing through and come out the other side. Whereas with active transport, you see how this closes up after the particle passes through? So when another particle was to come and bounce at this point, it would be closed. The membrane would be closed. And therefore the particle would be trapped on the inside of the cell, which is what the cell wants. Okay? But again, when we do osmosis and diffusion, we'll talk more about active and passive transport. This is just another picture where I'm showing you that protein molecule that basically opens up and allows things to go in and out through that lipid bilayer. Okay? After this, our next topic that we're going to be covering is going to be osmosis and diffusion. Okay?